So first of all, I'd like to thank for the great honor of being selected for this talk. And so today I'm going to tell you a short story about how temperature is measured in living organisms. So in everyday life, we know how to measure temperature, and one way to do that is to exploit thermal expansion of various materials, like mercury, for instance. If this is a droplet of mercury and at room temperature, and I heat it by 10 degrees, the droplet expands, and when I cool it back down, it shrinks again. And of course, you can't see that because the thermal expansion coefficient, beta, is very small. But I can enhance these motions using a trick. If I confine this droplet into a glass bulb, which constrains the expansion into a very thin diameter glass capillary, then the same temperature elevation will result in a very large change in the height of this thin mercury column. And so with that, I have greatly enhanced the sensitivity of this thermometer. On the other hand, if I cool the temperature now below 25 degrees, the mercury droplet will further shrink, but again, I can't see that. So clearly, this thermometer will have a temperature threshold of 25 degrees. And so sensitivity and threshold are two very important parameters of every thermometer. Now, we all have personal experience that our bodies can measure temperature, and one evidence for that is that if we touch something, we can immediately tell whether it is hot or cold. This is called peripheral temperature sensing. But another piece of evidence is that we are able to maintain our body temperature at a constant 37 degrees. And if our body temperature falls just a tiny bit, then this is immediately perceived by a specialized center in the brain. This is called central temperature sensing. And then physiological responses are initiated that ensure that at least the temperature in the core of our body remains near 37. And so this information between the periphery and the brain is communicated through nerve impulses, which are electrical impulses, and which are ultimately generated by ion channels that reside in the membranes of these nerve cells. So what are ion channels? Ion channels are transmembrane protein pores that can fluctuate between closed and open conformations. And when they are open, they are able to pass a certain type of ion, let's say a cation, through them. And this ion flux generates current, and it is ultimately this current that underlies all electrical nerve impulse propagation. So using suitable technology, one can measure the current that flows across such a tiny membrane area, which may contain just one such ion channel pore. And in such single channel current recordings, downward current deflections reflect inward current flowing through the pore. And in such recordings, one can clearly see the periods of time during which the pore is closed and the periods of time during which it is open. And this fluctuation between closed and open, called gating, is at equilibrium. It is characterized by an equilibrium constant. And in this specific case, you can see that the pore spends about half of its time closed and half of its time open, so the equilibrium constant is about 1. But that equilibrium constant is not static. It can be regulated. It can be increased, for instance, to very large values, so that the pore spends most of its time open, or it can be decreased to very small values, so that the pore spends most of its time closed. And this regulation is achieved in various different ways for different types of channels. In voltage-sensitive channels, there is a charged part of the protein which moves upward when the pore opens. And for such channels, this equilibrium constant is increased by membrane depolarization. And then there are ligand-sensitive channels which contain large cytosolic domains to which small diffusible molecules called ligands can bind. And for such channels, Kf is increased by ligand binding. But how can gating be regulated by temperature? It turns out that in some ion channels, pore opening is accompanied by breakage of a very large number of chemical bonds, like hydrogen bonds or ionic interactions. These are represented here by these little red ribbons. You can see there are many more such ribbons in the closed than in the open conformation. And then you might ask the question, if many bonds need to be broken for the pore to open, then why does such a pore ever open? And that happens because at the same time, the protein becomes more disordered upon opening. And these two natural tendencies of a system to prefer lower energy conformations on one hand, but more disordered conformations on the other hand, they balance each other out. And it turns out that for such a mechanism, pore opening will be promoted by heat, which provides the energy to break these bonds and also promotes disorder. And so for such channels, the Kf, equilibrium constant, will depend roughly exponentially on temperature. And that means if we plot Kf on a logarithmic scale as a function of temperature, we get a straight line. And the slope of this line, the sensitivity of this thermometer, will essentially depend on the number of chemical bonds that are broken during the pore opening process. Now, I've confined here the Kf axis to between 0.03 and 30. That's about the range of Kf values that are biologically decodable. And by that I mean, if Kf is as low as 0.03, that means the pore already spends most of its time closed, so any further decrease in Kf will not elicit any biological response. And conversely, if Kf is as large as 30, then the pore will be already open almost all the time, and any further increase in Kf will again not elicit a biological response. Now it turns out that the TRP or TRP family channels, they work just like that. TRP V1 and V2 are found in nerve endings in the skin, and they are responsible for detecting noxious heat, like when we touch a hot plate, for instance. And then there are also channels that have reverse temperature sensitivity, like TRPA1 or M8. In these, chemical bonds need to be broken when the pore closes, and these are involved in detecting noxious cold. And just a fun fact, many of these channels are also activated by ligand binding. So for instance, capsaicin, a compound present in chili pepper, activates TRPV channels in our taste buds, and thereby it fools us. It makes us believe that we are eating something very, very warm. And conversely, menthol, which is present in peppermint, activates TRPM8 channels in our taste buds, and we perceive it as if we were ingesting something cold. So these and many other TRP channels are involved in peripheral temperature sensing, but in contrast to our skin, the temperature of the brain is confined to a very, very narrow region, just about 1 degree, around 37 degrees, and even during very high fever, it rarely rises above 40 degrees. And based on these temperature activation curves, these channels are clearly not suitable to detect brain temperatures. And so it was just a few years ago that TRPM2 was discovered by a German group as being responsible for measuring temperatures in the brain. Inhibition of TRPM2 caused fever, and stimulation of TRPM2 caused body cooling in live animals. And that is where our group came into the picture, because we had been studying TRPM2 already for some time, and we knew quite a lot about it. For instance, we knew that it is a ligand-gated channel, which opens by binding two types of ligand, ADP ribose, a nucleotide, and calcium ions. But we also knew from structural studies done by cryo-electron microscopy that indeed upon pore opening there are many chemical bonds broken in this structure. You can appreciate that by comparing the closed and open conformations of this channel. There is a very large rearrangement in this cytoplasmic domain. And so we were interested, if TRPM2 is indeed involved in measuring body temperature, then by what mechanism? Is temperature dependence of TRPM2 an intrinsic property of the protein, or does it just reflect temperature-dependent changes in the concentrations of its ligands? Or are the ligands required at all, or is just heating in itself enough to open TRPM2 channels? And finally, is the threshold and sensitivity of TRPM2 optimal for measuring these tiny fluctuations around 37 degrees? So here you can see how TRPM2 currents are activated by simultaneous application of both ligands. This is current measured from a membrane area that contains hundreds of TRPM2 channels. So this very large current reflects the sum of those individual channel currents. And you can see when the ligands are washed away, the channels close. And so we first asked, what if we just heat the channels to 40 degrees? Will they open up? And you can see that they don't. And that is not because they would have been somehow destroyed by heating to 40 degrees, because if you now reapply the ligands at 40 degrees, we can again reversibly open and close the channels. So clearly, heating alone is insufficient to open TRPM2 channels. So we next asked, is perhaps heating plus the presence of just one of the ligands sufficient? What if we apply just ADP ribose combined with heating? And applying ADP ribose alone at room temperature provides a tiny little current. Is that enhanced at 40 degrees? Well, it is to some extent. If I zoom in on this part of the record, you can now clearly see individual channel openings. But if you compare that current to the activity that can be elicited by exposure to both ligands, you can immediately see that that activity is still very, very small. And so if we quantitate this Kf at the two temperatures, we get this temperature sensitivity curve in the presence of saturating ADP ribose, which is clearly incompatible with measuring temperature in the brain. So how about the other ligand, just calcium? Is just calcium plus heating enough? And the result is very similar. Maybe we get a tiny bit more current at 40 degrees with just calcium, but this temperature activation curve is still incompatible with the brain. So the conclusion is, even with one ligand present at saturating concentration, the temperature threshold of TRPM2 is too high. So then we asked, how is the current elicited by the presence of both ligands at saturating concentrations modulated by temperature? And you can see that such currents indeed get smaller upon cooling, but increase upon heating. But if we now quantitate Kf at 37 degrees, it turns out that at 37 degrees under such conditions, the channels are already almost fully open. And so in that case, looking at that temperature activation curve, we find that the temperature threshold is now too low. And so we reason, because the presence of ligands shifts this temperature activation curve to the left, there must exist some intermediate subsaturating concentration of ligands that shifts the temperature threshold to just an optimal range. And why? Well, if the ligands are present at subsaturating concentrations, that means the binding sites will spend part of the time ligand bound and part of the time empty, and so you will expect to see some kind of an average behavior between these two extreme behaviors. And moreover, the channel is four-fold symmetric. It contains 4 ADP ribose binding sites and 4 calcium binding sites. So there are altogether 25 patterns of possible ligand binding. You can have 0, 1, 2, 3, or 4 ADP ribose molecules bound with 0, 1, 2, 3, or 4 calcium ions bound. And all of these configurations are able to open up, but I've shown here the open states only for the back row for reasons of clarity. So we first fixed ADP ribose concentration to a saturating level and varied the calcium concentration. By that, we essentially restrict gating to this subset of the model. And we measured channel behavior at 3 different temperatures between 14 and 40 degrees. And then we did the opposite. We fixed calcium to saturating levels and altered the ADP ribose concentrations. And again, here are the measured activities at those 3 temperatures. And then by fitting this model to this data set, we could estimate all the parameters of this model. And you can see that this model provides quite a good fit to this data. And that means that using the model, we can now predict channel activity at any calcium concentration, any ADP ribose concentration, and any temperature. And so from such calculations, we could tell that it takes low micromolar concentrations of both ligands to shift the temperature sensing curve into an optimal range. And that is good news and bad news. It is good news because for ADP ribose, low micromolar is a typical cytosolic concentration. But it is bad news because for calcium, a typical cytosolic concentration is about 50 times lower. It's only about 0.1 micromolar. And so the last question was, in a living cell, where does such a high calcium concentration come from? And to understand that, I need to tell you a little more about the structure of the channel. So this cartoon is a cross-section of the channel structure, and the ADP ribose binding site is found in this large cytosolic domain, but the calcium binding sites are right here at the membrane-cytosol interface, very near the pore. Now, in all the experiments I've shown you so far, we have supplied both of the ligands from the cytosolic side, and in particular, we have kept external calcium at 0. And we have seen that under such conditions, at a low 0.1 micromolar cytosolic calcium concentration, which is typical for a cell, the channels do not open in the physiological temperature range. However, in a living organism, external calcium is 10,000 times higher. It is millimolar. And moreover, we know that the TRPM2 pore itself conducts calcium ions. And so we reason that perhaps calcium influx through the pore generates a local calcium cloud here, in which the calcium concentration becomes high enough. And indeed, when we measured currents under such physiological conditions at 37 degrees, in the presence of ADP ribose and with 0.1 micromolar bulk cytosolic but 1 millimolar external calcium, we could indeed record a channel activity. And that is a small activity, but it is clearly quantifiable. And then using the model, we can ask what must have been the local calcium concentration to ensure that level of activity. And if we do that, we come up with a value of about 2 micromolar, which is about 20 times higher than the bulk cytosolic value. And if we now measure currents under identical conditions but at 40 degrees, we get very robust activity, a Kf of about 0.5. And if you now connect these 2 points, you can clearly see that that temperature activation curve is much steeper than what you can get with any fixed ligand concentrations. And that is a clear indication that at 40 degrees, local calcium concentration has risen to even higher values. And if we back calculate that value from the model, we now come up with a value of about 4 micromolar. So there is clearly a positive feedback mechanism operating here. At higher temperatures, the pore spends a larger fraction of time open, so more calcium flows in, local calcium concentration rises even higher, and that feeds back on channel gating by further stimulating channel activity. And it is due to this positive feedback mechanism that TRPM2 becomes extremely temperature sensitive, so that its Kx value increases by greater than 10-fold over a range of just 3 degrees Celsius, as required for a deep brain temperature sensor. So in summary, I've shown you that TRPM2 gating is intrinsically temperature sensitive, but its threshold is too high with 0 or 1 ligand bound, but too low with both ligands present at saturating concentrations. The threshold is around 37 degrees at physiological ADP ribose concentrations, but higher than physiological calcium concentrations. That high calcium concentration is provided by calcium flowing in through the channel's own pore. And finally, that calcium influx also provides a positive feedback loop, which endows TRPM2 with an extreme temperature sensitivity. And so with that, let me end by thanking the team, the group who has done the work, also agencies that have provided funding, and especially my colleague Adam Bartok, who has done all the recordings I've shown you today.